Hi everyone, it's Dr. LeBlanc with Unit 11B, Variations on Mendelian Inheritance. Our learning goals are to understand how simple Mendelian inheritance can be altered, to understand the concepts of wild type and norm of reaction, to understand haploinsufficient, loss of function, gain of function, and dominant negative alleles in relation to mutant phenotypes, to understand the concept of incomplete dominance, to understand the concepts of incomplete penetrance and variable expressivity, and to understand how pleiotropy, including lethality, epistasis, complementation, and gene interactions can influence the outcomes of genetic crosses. Sometimes, when you mix a recessive allele and a dominant allele, you don't get the dominant phenotype, but you get an intermediate phenotype instead. Simple Mendelian inheritance has a few definitions that we need to go over before we deviate from those. First of all, wild-type alleles are the most prevalent alleles within a population. There can be more than one. That indicates a genetic polymorphism. They're made usually in the right amounts, the correct amounts, and they are typically dominant, Mutant alleles differ from wild-type and tend to be rare in natural populations. They can be recessive, such as loss-of-function alleles, in which a protein is inactive or reduced in amount. They can be dominant. They could function better than the wild type. That's called a gain of function. They could antagonize the wild type allele, and that's a dominant negative function. Or they could be haploinsufficient, which means that loss of function of one allele doesn't have enough of the protein to exhibit the wild type trait. Okay, so what are some of the variations on Mendelian inheritance? Well, two of them are incomplete penetrance and variable expressivity. This gentleman in the photo has a genetic condition. Can you tell what it is? Well, if you count 1, 2, 3, 4, 5, this condition is called polydactyly. It's caused by a dominant allele. However, this condition exhibits incomplete penetrance, which means that individuals that inherit the dominant allele sometimes do not express it. So, if this is a dominant allele, this dad is passing it on to all of his children here, or some of them, and they can pass it on to their children. Okay? Now, here's an indication where if one of these has a dominant allele, it's not showing it, but one of their children, two of their children do show it. Dominant alleles with incomplete penetrance can be tricky to interpret from a pedigree. This individual not only is showing a gene that has incomplete penetrance, okay, in this individual, the gene did penetrate because he does have an extra finger, but it also exhibits variable expressivity, which means that sometimes the finger can be a complete finger and sometimes it can be a very short little stubby finger. Here's another example of variable expressivity. Okay. The piebald gene is a gene that's expressed in beagles. Every single one of these beagles is expressing the piebald gene. So we say that penetrance is 100% in this population. However, the expressivity varies greatly. In some instances, it's a very low expressivity, and some it's very high. Each dog shown is heterozygous for the piebald allele. There can also be environmental influence on the expression of a genotype. A norm of reaction or a phenotypic range is exhibited under different environmental conditions. This is the same Arctic fox in winter and in summer. No genetic difference between these two foxes. The only difference is the environment. The norm of reaction for an eye facet number in genetically identical flies raised at different temperatures shows that at low temperatures, the facet number is high, but as the temperature increases, the number of eye facets decreases. That's the norm of reaction. That's what normally happens in a fly. There's no genetic difference that is influencing the number of facets. The only thing that's influencing them is the temperature. Okay, so we have a red flower and a white flower, and what do we end up with? A pink flower. How do we explain that? Incomplete dominance is when a heterozygote individual exhibits an intermediate phenotype. So, intermediate between red and white is pink. That's pretty obvious. If we assign genotypes to these individuals, this true breeding red flower has a color red and a color red allele, and this one has a color white and a color white allele. They each produce one kind of gamete each, a red and a white, and when you mix those together, you get a pink flower with a CRCW genotype for that particular gene. If you take a heterozygous individual and self-cross it, then you can end up with an interesting ratio in the F2 generation. Both the sperm and the eggs can be produced with either the CR allele or the CW allele. Two CR alleles are going to give you a red flower. Two CW alleles are going to give you a white flower, but intermediate in between that, a CR and a CW allele are going to give you a pink flower. So our ratio in the F2 generation is a 1 to 2 to 1 ratio. Sometimes there can be many more than 2 alleles for a single gene within a population, and they all may be equivalent. This is the glycocalyx of a red blood cell membrane. Here's the membrane here, and this is the sugar coating that exists on the outer surface of a red blood cell. Here's a red blood cell, and here is a branched sugar that exists on its surface. Okay, it has several sugars here that can be modified by an enzyme called glycosyltransferase. Now, glycosyltransferase comes in 3 different varieties. Glycosyltransferase encoded by the IA allele will take N-acetylglycosamine and attach it to one of the basic sugars on the coating of the red blood cell. When it does that, it creates an antigen called A. It's an A antigen. That's the unbranched form of the sugar on the surface of the red blood cell is called antigen O. Now, if you have a different allele of the glycosyltransferase called IB, it will take galactose and attach it to the basic sugar on the red blood cell and create something called antigen B. So far, we have 2 different alleles of the glycosyltransferase, one that attaches N-acetylgalactosamine and one that attaches galactose. There's a third allele that is inactive, or it's defective, glycosyltransferase, and it leaves the sugars unmodified, creating the O antigen. So we have 3 different possibilities for how the sugars on the red blood cell are modified. No modification, galactosamine, and galactose. That yields all the different types of blood that are possible. So remember that the I allele, the little I allele, is the defective glycosyltransferase, leaving the sugars unmodified. The other alleles, the big IA allele or the big IB allele, encode the other types of glycosyltransferase. So, what are our possibilities? Well, we could have red blood cells that have produced from individuals with the two recessive alleles, the inactive form of the glycosyltransferase. They have only unmodified sugars on their surface, or antigen O. We could have individuals that have only the A antigen. They could be produced by either individuals that are homozygous for the A allele for the glycosyltransferase, or heterozygous. In either case, for either genome, either genotype, you're going to have a sugar that has only A modification. If you have two IB alleles or an IB and a little I allele, then you're going to produce red blood cells with only the B antigen. And if you have a glycosyltransferase that adds an A and a glycosyltransferase that adds a B, you can have an AB phenotype. So that is how you end up with the 4 different blood groups for humans. Let's see what happens if an individual that is heterozygous for the A glycosyltransferase and another marries and has children with another individual that has the IB allele and is also heterozygous. So the sperm can be either IB or little I, and the eggs can be IA or little I. And so what happens? Their children could be produced in equal percentages, type AB, type A, type B, or type O. What about some lethal alleles? What happens to the ratio of offspring that are produced when you have a lethal allele? This is a Manx cat. It has a very short stubby tail. The Manx allele is dominant. So if you inherit one of the Manx alleles, big M, you end up with a cat that has this short stubby tail. Okay? If you cross one Manx cat with another Manx cat, you'll end up with these type of sperm and these type of eggs. And when you fill in the Punnett square, you end up with a 1 to 2 to 1 genotypic ratio. However, if you inherit 2 of the dominant Manx alleles, you end up dying as an early embryo. So kittens are born with the Manx phenotype in a 1 to 2 ratio or 2 to 1 ratio because these kittens are never born. Okay? So you'll end up with 2 Manx cats for every non-Manx cat in a cross between 2 Manx cats. Pleiotropic effects. When you have multiple effects by a single gene on the phenotype of an organism, it is called pleiotropy. When you have pleiotropic effects, it's usually because that same defective protein may be expressed in multiple tissues at different stages of development, multiple stages of development, and it may be used by the cell in different ways. Our example of pleiotropy is cystic fibrosis. The normal allele encodes a transmembrane conductance regulator, so it's called the the cystic fibrosis transmembrane conductance regulator. It lets chloride ions cross the membrane. The mutant does not transport chloride ions effectively. This has widespread effects on the individual because the CFTR is expressed in many tissues. In lungs, it causes very thick mucus that can cause multiple infections. On the skin, it can cause a very salty sweat. And males are often sterile because the CO transport is needed for proper development of the vas deferens. Thus, the CFTR can have multiple or pleiotropic effects. Okay, let's look at another effect, complementation. Complementation is a phenomenon in which two different parents that express the same or similar recessive phenotypes produce offspring with a wild-type phenotype. How is that possible? So here we have a cross involving a two-gene interaction. What's a two-gene interaction? Well, if you have a phenotype that is the product of two genes that interact, that is a two-gene interaction. Since you have two genes that are necessary to produce the phenotype, you could have mutations in one or the other of those two genes. So let's take our example. We have a white flower from one meadow, and across the valley we have some white flowers that are very similar, look exactly the same, but they're distinct from this population is distinct from this population. Okay. When you take those back to the greenhouse and cross them, you will find that you end up with purple flowers. How can that be? Well, you surmise that the purple pigmentation in this flower is the product of a pathway, a biochemical pathway. So you start with a colorless precursor, and you turn that into a colorless intermediate, and then you turn that colorless intermediate into a purple pigment. And these steps each require their own enzymes, which are the product of two distinct genes. So you have enzyme C, which will convert the colorless precursor into a colorless intermediate. And you have enzyme P that will take the colorless intermediate and turn it into a purple pigment. So if we go back to our cross and assign some genotypes to these flowers, this true breeding strain of white variety number 1, we'll say, has two good C alleles, but the P alleles for enzyme P are mutant, both of them. So they're homozygous for the mutant P allele recessive, and they are homozygous dominant for enzyme C. So they're making lots of good enzyme C, but no good enzyme P. All the enzyme P they make is defective. And the reciprocal is true for the white flowers that we found across the valley. These are making defective enzyme C, but their enzyme P is just fine. But neither one of these can convert anything into the purple pigment because you need both of these enzymes to be active in this pathway in order to get the purple pigment. When you cross these together, you end up with the F1 generation that is heterozygous at both genes. It has a dominant allele for the C gene from this flower, and it has a dominant P allele from this flower. So this flower has a good enzyme C and a good enzyme P, so it's fully capable of completing this entire pathway, producing the purple pigment. That is called complementation. Each recessive allele, the C and the P, is complemented by a wild-type allele. This phenomenon indicates that the recessive alleles are in different genes. Okay, so what happens if you take this flower and self-fertilize and raise some plants of the F2 generation? What you end up with is a ratio that is not 9 to 3 to 3 to 1. You end up with either purple or white flowers. Epistasis shows that homozygosity for the recessive allele of either gene results in the white phenotype, and that masks the purple phenotype. So both gene products encoded by the wild type alleles are needed for the purple phenotype. So epistasis is when one gene product affects the genotype of the other gene product. So, for example, here's an individual that has two good P alleles, but because it has two recessive C alleles, you can't see the effect of these two good P alleles. That is epistasis, when one gene masks the effect of the other. Mendelian inheritance can be altered in a variety of ways, including environmental conditions and interactions between genes. Wild type is the most prevalent phenotype in a population. How that phenotype can be altered under variable environmental conditions is called the norm of reaction. Haploinsufficient is a dominant mutation where one good copy of the gene is not sufficient to produce a wild-type phenotype. Loss of function is a recessive mutation where if both copies of a mutant gene are inherited, the individual expresses the mutant phenotype. That's the normal recessive condition. Gain of function means that a mutation alters a protein so that it has a new or increased level activity. It is dominant. Dominant negative means that a mutation that causes a deleterious effect need only be present in one copy to express the phenotype. It is dominant. Sometimes that's referred to as a poison product if it is that particular type of dominant negative mutation. Incomplete dominance is exhibited when two different alleles express themselves simultaneously and do not influence the phenotype of the other. That's what we see in the blood type groups, especially in the AB blood type. Incomplete penetrance happens when individuals who carry a gene do not express it phenotypically. That's typically seen with polydactyly. People who have that dominant allele sometimes do not express that extra digit either on their toes or on their hands. Variable expressivity occurs when a gene is expressed to a greater or lesser degree by different individuals within a population. All those piebald dogs looked quite different, and that's variable expressivity. Pleiotropy occurs when one mutation affects many different phenotypes simultaneously, such as the cystic fibrosis gene. Epistasis is when one gene mutation affects the expression of the other, such as the two white flowers repressing the effect of a dominant homozygous for the dominant allele of the other gene. Complementation occurs when two or more gene products are necessary to produce a phenotype.